Hello everyone, my name is Kiran Chaudhary and I am your biology faculty at Infinity Learn by Sri Chaitanya. I welcome you all in today's session, students, where I will be discussing about one very important topic of your chapter number 6 of 12th class NCERT biology textbook. The chapter name is Molecular Basis of Inheritance, and in this class we will discuss about transcription. Let me also tell you, students, in this session we will take introduction about transcription process. That means what is transcription and what is a transcription unit that actually participate in this process. So, that is the objective of today's session. I hope this is clear to all of you. Now, let us start the session, and before starting, obviously I would love to give you important information that please visit to infinitylearn.com website and check out the courses and subscription that we offer there. You can use those courses, you can use those subscription at 40% off by using the code Kiran40. So, please check out the courses that we offer at infinitylearn.com and use the code Kiran40 to get them at 40% off. Let us start the session now. See, students, what is transcription? We will start with the definition itself. Transcription is a process in which genetic information will be copied from DNA to RNA. Very, very important, right? So, it is a process. Transcription is a process where genetic information को copy किया जाएगा कहाँ पर? RNA के अंदर, right? I hope this much is clear. Second thing that you need to understand is that only one of the strand of the DNA will participate in transcription process. Yes, it is not like DNA, it is not like replication process of DNA. You remember I taught you in the previous class about replication process. In replication process, what was happening? The genetic information from both the strands of the DNA was getting copied, was getting transferred to another molecule of DNA. So, in replication process, from one DNA molecule, another DNA molecule was getting created first of all, and second, both the strands were taking part. But when we talk about transcription process, only one strand of the DNA molecule will participate, and secondly, genetic information from DNA will be copied to RNA, right? I hope this much is clear to all of you, and this is exactly what the first line says. Process of copying genetic information from one strand, only one strand will participate of the DNA into RNA. Next, students, while copying this genetic information from DNA to RNA, complementary rules will be used. Remember complementarity rules that I have already taught you in the few initial classes of this chapter. So, what will happen? Complementarity rule will be followed while copying this information. And what is this complementarity rule? Remember, purines will pair with pyrimidines, right? Purines will pair with pyrimidine, and in the purine also I can tell you which purines and pyrimidines will participate in this process. See, in transcription process, adenine will actually pair with uracil, right? This is something different from replication process. Remember, my kids, during replication process, adenine used to pair with thymine with the help of 2 hydrogen bonds. But here in this transcription process, in place of thymine, uracil will be used, right? Second, and vice versa is also followed. Uracil will pair with adenine with the help of 2 hydrogen bonds. Second, guanine will pair with cytosine with the help of 3 hydrogen bond, and reverse, cytosine will be paired with the help of 3 hydrogen bonds with guanine. So, this is what going to happen, in complementarity, right. So, this pairing will take place when genetic information from one strand of the DNA will be copied on to an RNA molecule, right. Secondly, very important thing, students, only a segment of DNA will participate. This is one of the important difference between replication and transcription. Remember, in transcription process, entire length of the DNA, for example, if we talk about human cell, 2.2 meter length of the DNA used to participate in replication process. But unlike replication, in transcription process, only a segment of DNA will participate, and from that segment also only one strand will participate. See, what is segment of DNA? This segment of DNA will possess what? Segment of DNA will possess two strands, and from those two strands only, only one of the strand will be copied, right? Remember, listen this very carefully, only and only from one strand of this segment of DNA, genetic information will be copied from DNA to RNA. So, please understand and mark this difference between replication and transcription. In replication, entire length of the DNA will participate, both the strands will participate. In transcription process, only a segment of DNA will participate, and from that segment also, that segment possesses two strands. So, from this segment also only one strand, genetic information present on one strand of this segment will be copied into an RNA molecule. I hope all these points are crystal clear to all of you. Now, let us talk about this very important question, right? So, remember here what I told you, only one of the strands is copied into an RNA molecule. So, this line should obviously spark one question in all of your mind, and that is why? Why both the strands are not copied? Why both the strands are not copied during transcription process? Why only one? Right? First of all, only a segment is participating. From that segment also of the DNA, only one strand is participating in transcription process. Why? This is an important question everybody should ask, right? Now, let us understand why this happened. To understand why this happened, we need to consider the opposite case. So, for a moment, let us understand or let us consider that both the strand, what if both the strand participate? So, from this point, I will take you to an explanation where I will explain you why only one strand participate. So, for now, let us consider that both the strands are participating, okay. So, see students, what is this? These are the two strands of that segment of DNA which is participating in the transcription process, right. So, let me mark it 5 dash, 3 dash, 3 dash and 5 dash. So, these are the two strands of the segment of DNA. Now, students, please try to understand this very important point. Both the strands of the DNA are not identical to each other in terms of sequence of nucleotides, right. These two strands are actually complementary to each other. They are not identical, they are complementary to each other with respect to the nucleotide sequence on them. And that also talking specifically about the nitrogen bases, right, because that is the one which is making the difference, right. Now, if both the strands are complementary to each other, now let us consider both are participating in transcription process. What will happen? If this strand is participating in transcription, process, then that means RNA will be formed from this molecule. For example, this one is participating and it is producing an mRNA, right. This strand, so when this strand will participate in transcription process, this will also produce an mRNA, right, this will also produce an mRNA. But now another question on which I want everybody to think is, what do you think, this mRNA and this mRNA, will they going to be identical to each other or they will be complementary? I want everybody to think on this question. Very simple answer students, both these mRNA will actually be complementary to each other. Why? Because they are individually formed from the two strands which are complementary to each other. When the strands of the DNA are not identical to each other, how come the mRNA that is produced from both these strands will be identical, right? Hence, these two mRNA won't be same in terms of the sequence of the nucleotides, but they will be complementary to each other, right? So, both these mRNA are not identical. They are not identical. Now, let us consider this mRNA and this mRNA. I am writing it as mRNA 1 and mRNA 2 because they are not identical, they are complementary. Remember, note, please note mRNA 1 and mRNA 2 will be complementary to each other. Right? Now, next students. So, let us consider this mRNA and this mRNA 2 both are participating in translation process. What will happen during translation process? Proteins will be formed according to the sequence of nucleotide that are present in these respective individual mRNAs. According to the sequence of the nucleotides, right, the sequence of the nucleotides present in the respective mRNA will form genetic code, and according to that tRNA will bring what? tRNA will bring amino acids, and hence according to the sequence of nucleotide present in this mRNA and this mRNA, the sequence of amino acids in the forming proteins will be decided, right. So, tell me, if this mRNA participates in translation process, it will form a protein, and if this mRNA also participates in translation process, this will also form protein, right. This will be protein 1, this will be protein 2. So, my lovely kids, please notice both these proteins are also not identical, and it is very obvious to guess that. Why? Because this protein and this protein, they are not formed from the identical or same mRNA molecule. When mRNA molecule from which these proteins are synthesized are complementary, hence the proteins will also not be same. Because of the complementarity nature, right, because mRNA 1 and mRNA 2 both are not identical, and hence they are complementary. Because of this fact, sequence of amino acids present in protein 1 and protein 2 will be different, right. Now, listen, carefully observe, if such thing happens, if both the strands of the segment of the DNA participate in transcription process, what are you observing? You are actually seeing formation of two different proteins, protein 1 and protein 2, formation of two different proteins from the same segment of DNA, and if this is going to happen, this thing will actually complicate the genetic material transfer machinery, and this is something that is not desirable, right. And secondly, if you notice, both mRNA 1 and mRNA 2 are not identical, they are complementary. Hence, if mRNA 1 and mRNA 2 will be complementary, there is possibility that mRNA 1 and mRNA 2 are going to form double-stranded RNA molecule, and if these two, mRNA 1 and mRNA 2, forms a double-stranded RNA molecule, then you are pretty smart to imagine that this double-stranded mRNA molecule will prevent the translation process, and the whole exercise of the cell doing transcription process and forming the mRNA will become waste, all will go in vain, right, because proteins won't be formed, because translation will be prevented. Why? Because of the formation of a double-stranded RNA molecules. When mRNA molecule won't be exposed, hence translation cannot take place over it. I hope now you all understood the two important reasons because of which both the strands of a segment of a DNA do not participate in transcription process. It is only one strand that participate in transcription process, right. So, these are the two reasons that I have already explained to you in a proper detail, right. Let us see the next thing now. Okay students, now let us discuss in a little detail about the transcription unit. What is a transcription unit? Transcription unit is that part or segment of DNA that actually participates in the transcription process. This transcription unit is made up of three important parts: promoter region, structural gene and terminator region. So, promoter, structural gene and terminator forms the parts of a structural unit that actually participates in transcription process. And remember, promoter, structural gene, terminator, all are what? They all are part of DNA, right? They all are part of DNA, part of a segment of DNA, right? So, they all are DNA in chemical nature. Do not forget that. So, what is transcription unit? Simply segment of DNA, part of DNA that is participating or taking part in transcription process. And second, it has three important regions. It is marked primarily by three important regions, right. First is promoter, structural gene and terminator. See, with this beautiful and understand, this image itself, like you can understand by your own self that what exactly are the three regions and how they are present in a transcription unit. So, this diagram actually represents two transcription unit where you can see a promoter, where you can see structural gene, where you can see terminator. Remember all three, promoter, structural gene and terminator are part of DNA, all are DNA in nature, right. Now, let us discuss in little detail about each one, right. We will start with the structural gene and then come on promoter and terminator, right. See students, if we talk about structural gene, see this is the structural gene. Remember students, this structural gene, this entire part is structural gene, right. And this structural gene is made up of two strands. Structural gene has template strand, structural gene has coding strand. Structural gene is a part of DNA and DNA is double stranded. And if I am saying structural gene is present on DNA, that means structural gene will possess two strands. So, structural gene has two strands. One is known as template strand, another is known as coding strand. So, that is the point. Let us try to understand why one of the strand of structural gene is known as coding and another is known as template strand. Why this difference? Okay. Now, before making you understand that, please try to understand that structural gene actually possess those important sequences of nucleotide using which RNA molecules will be produced. So, this is the structural gene that will participate in the transcription process. Please do not forget this point, right. This is the most important region of our transcription unit, right. So, see students, structural gene, template and coding. Now, let us understand why such. So, see students, the main enzyme that participate in the transcription unit is RNA polymerase. Rather, I should say DNA dependent RNA polymerase. Why the name so? Because this RNA polymerase will polymerize the ribonucleotides in order to form RNA molecule in template dependent fashion. It will utilize one of the strands of the DNA as a template on which, according to the sequence of the nucleotides on that particular template strand of the DNA, with the help of this DNA dependent RNA polymerase will polymerize the ribonucleotide to form RNA, hence the name DNA dependent RNA polymerase. Now, there is again important special characteristic about this RNA polymerase is it can polymerize the ribonucleotide only in one particular fashion or direction, that is 5:3. It cannot do reverse. So, it will polymerize the ribonucleotide in 5:3 direction only. Hence, because of this, one of the strands will become or act as a template of structural gene. Because of this activity of RNA polymerase, that it can polymerize only in direction 5:3, hence making one of the strand of structural gene as template and another as non-template one, also known as coding strand. So, you are pretty smart to guess if RNA polymerase is polymerizing in 5:3 direction, that means the strand of the structural gene which has the exact opposite polarity 3:5 will act as a template in the transcription process on which this RNA polymerase will use that strand as a template and polymerize the ribonucleotides, right. So, hence 5:3 is the direction in which RNA polymerase is polymerizing, because of which the strand of the structural gene which possess the polarity 3:5 becomes template for this RNA polymerase in this transcription process, right. And the strand of the structural gene that possess the polarity 5:3 becomes non-template, also known as coding strand. So, by default these names are given and you need to consider that, right. Coding strand will actually not code for anything, right, but by default all the directions or all the naming as well as the positioning of the positioning of the promoter and terminator will be done with respect to the coding strand, right. So, I hope this much is clear to all of you. Now, let us see on the board as well. The strand which possess the polarity. So, we are talking about structural gene. Structural gene, this is the template strand, this is the coding strand. Template strand is having the polarity 3:5. So, why the strand 3:5 termed as template? Because of the limitation of RNA polymerase, that it can polymerize in 5:3 direction. And the strand of the structural gene that possess the polarity 5:3 will act as a coding strand, and this coding strand will not participate, does not code for anything, right. It won't code for any mRNA molecule, right. So, this is exactly what I was trying to explain you. What does template strand means? That will participate in transcription. And what does coding strand means? This coding strand won't participate in transcription process. But there is one very important fact that you need to remember is this coding strand has the sequence same as that of the RNA except T at the place of U. Very important fact, and in fact on this fact you can see multiple MCQs that NTA has asked in NEET exam. Very important. So, please remember, although this coding strand won't participate in transcription process, but the RNA molecule that is synthesized during transcription process using the template strand, the sequence of the nucleotides present in the RNA molecule will be similar to the one that is present, will be similar to the nucleotide sequence that is present in the coding strand, except one difference, that since DNA is going to have thymine, and in place of thymine, RNA molecule will possess uracil, right. So, just remember, coding strand, sequence of nucleotides present in coding strand will be same as that of the nucleotide sequence present in RNA molecule, except one difference. In coding strand you will see T, and in place of T in RNA molecule you will find U. So, please remember this fact, and on this we will solve one question also on the very next slide. Very important fact, students, all the reference points while defining a transcription unit is made with the help of coding strand. So, coding strand, actually the polarity of the coding strand is used to assign the position of promoter, terminator and other segments or parts of transcription unit. So, coding strand by default is used for reference, for giving positions to the segments or the parts or forms of structural unit. I hope this much is clear to all of you. Good evening everybody. Okay. Next, what we need to understand, this is an important question given in your NCERT as well. This is a hypothetical sequence of the transcription unit. This is the template strand which will actually participate in transcription, right? It will actually participate in transcription. This is the coding strand. Remember, it won't code for anything, but remember this fact, remember this fact, right? So, sequence coding strand has the same sequence as that of the RNA. So, can you now write the sequence of RNA transcribed from the above DNA? Remember, template strand will participate in transcription process and the formed RNA, sequence of nucleotide formed in the RNA from this template strand will be same as that of the sequence of nucleotide present in the coding strand, except wherever there is T in RNA, there will be, wherever there is T, this one T, there will be U, wherever T, there will be U, wherever T, there will be U, and A will actually pair with C, wherever T, there will be U, right. So, I hope this much is clear. So, for forming RNA molecule, A will pair with U and G will pair with C. So, I hope this much is clear to all of you. So, in place of T, you have to imagine, in place of U, let me write it properly for all of you. I think some of you might get confused over here. So, wherever T, so this rule, you need to follow this rule, and you know who pairs with T in place, who pairs with T when it is a DNA molecule, adenine, A and T pairs with each other. So, in place of T, who will come? In place of T, A will come. In place of A, opposite to that, who will come? Uracil. So, opposite to C, who will come? G. Opposite to G, who will come? C. Opposite to T, who will come? Opposite to T, A will come, and opposite to A, who will come? U will come, right. So, I hope like this you can complete it, right. So, I hope this much is clear to all of you. Chaliye, next what we need to understand is this. Let us discuss in little detail about promoter and terminator regions as well. First of all, please remember this very important fact. That I want everybody to understand is that first we will see how the position of promoter and terminator is defined using coding strand of the structural gene, and second, what is the function of promoter and terminator. So, remember what you need to use? You need to use coding strand of the structural gene. So, see, this is the coding strand of the structural gene. So, promoter is present on, this is the coding, you need to focus on this green strand. So, from this coding strand, if you see, promoter is present at 5 dash and it is present ahead of, promoter is present ahead of the structural gene or coding strand. Hence, the promoter position is 5 dash upstream. So, promoter has 5, it is present at 5 end upstream of the structural gene. In that, you need to consider coding strand for all the reference points. Now, next see this. This terminator is present at the 3 dash end of the coding strand of the structural gene, as well as you can say that this terminator is present at the back of the coding strand, hence the name given downstream. So, let us talk about the position. Terminator is present at the 3' end downstream, means at the back of the coding strand of the structural gene. So, according to the coding strand, Next, let us talk about the function. The promoter actually possesses those sequences of the nucleotides of DNA at which RNA polymerase will come and bind and initiate the transcription process. So, DNA sequence providing binding site to RNA polymerase. Promoter will possess binding site, will provide binding site to RNA polymerase, where RNA polymerase can come, identify and start the transcription process. And terminator usually marks or defines the end of the transcription process. As the name suggests, terminator will terminate the transcription process. So, that is all students I wanted to discuss in today's session. Remember what we did, we understood or we took the introduction about transcription process, and we also understood what is a transcription unit. It is made up of what things which actually participate in transcription process. So, that is all from today's session students. Thank you for joining my session. Please give me a thumbs up if you like me and this session, and do not forget to subscribe Infinity Learn by Sri Chaitanya. Also hit the bell icon so that you will be notified every time whenever I will go live, and you won't miss any session in future, right? And also do not forget to give me proper comments that will help me to improve on my coming sessions. Thank you students. That's all from today's class.